Hey guys, this is the last section for Module 5, The Chemistry of Life. I'm on page 148, Lipids. Ready? Let's go. Lipids. Lipids, also known as fats, are complex molecules formed when three fatty acids, like the ones shown in figure 5.5, link to a substance known as glycerol in a dehydration reaction. A simple dehydration reaction that forms a lipid is shown in the figure below. In general, lipids cannot be dissolved in water. Cooking oil, for example, is made of lipids. What happens when you try to mix water and cooking oil? They don't mix well, do they? This is because the structure and components of lipid molecules make them hydrophobic. Hydrophobic, lacking any affinity to water. Since lipids are not attracted to water, they simply cannot be dissolved in it. Lipids are important biologically because most animals store most of their excess foods as lipids. Even though animals can take their excess carbohydrates and turn them into glycogen, as we learned just a few pages ago, lipids are the preferred method of storing excess food because more than twice as much energy can be stored in an ounce of fat than in an ounce of carbohydrates. As a result, animals make fats whenever they have excess food. Later, when food is scarce, their bodies can digest the fat for energy. For most humans in the United States, of course, there isn't a lot of food scarcity. Thus, the fat just builds up in our bodies, making us overweight. The fact that more than twice as much energy can be stored in an ounce of fat as can be stored in an ounce of carbohydrates has implications for our diets. People tend to like the taste of fats, and in the United States, we have a lot of food that is rich in fat. Since a lot of energy can be stored in fat, the higher your diet is in fat, the more excess energy you tend to intake. After all, most people tend to eat until their stomachs are full. Well, if you fill your stomach with foods that are mostly fat, you have taken in twice as much energy as you would have if you had filled your stomach with foods that are mostly carbohydrates. As a result, your body will have a lot more excess energy, and it will make more fat to store that excess energy. Diets high in fat, then, can lead to being overweight. Nowadays, nutritionists also tell us that saturated fats tend to lead to heart troubles, while unsaturated fats are less likely to. As a result, they suggest that we eat unsaturated fats. What's the difference? When a fat is made from fatty acids that have no double bonds between the carbons, it forms a saturated fat. We call the fat saturated because it has all of the hydrogens it can take. As a result, it is saturated with hydrogen. If a fatty acid has one or more double bonds between the carbons, however, it has fewer hydrogens than it possibly could. As a result, we call it unsaturated. Saturated fat, a lipid made from fatty acids that have no double bonds between carbon atoms. Unsaturated fat, a lipid made from fatty acids that have at least one double bond between carbon atoms. Look back at figure 5.7. The fatty acid molecules that make the fat drawn in that diagram are saturated. as there are no double bonds between the carbon atoms. When saturated fatty acid molecules attach to the glycerol, they make a saturated fat. If, on the other hand, there are one or more double bonds between the carbons in the fatty acid molecules, when they link to the glycerol, they make an unsaturated fat. How do you know whether a fat is saturated or unsaturated? Well, one way is to look at the structural formula of the fat. If there are double bonds between the carbons in the fatty acids, it is an unsaturated fat. Otherwise, it is saturated. There is another way to distinguish these two types of fats, however. Generally, you can look at their phase. At room temperature, saturated fats tend to be solid, while unsaturated fats tend to be liquid. Thus, the solid lard that you use for baking is a saturated fat. The liquid oils that you use for frying, however, are unsaturated fats. Proteins and enzymes. Proteins make up one of the most important groups of molecules in the chemistry of life. They are involved in virtually every chemical reaction that supports life. As you might expect, they come in many different shapes and sizes and are very complex. They are mostly made from carbon, nitrogen, hydrogen, and oxygen, but some proteins have phosphorus and sulfur in them as well. Like polysaccharides, proteins have some basic building blocks. The basic building blocks of a protein are called amino acids. There are about 20 different amino acids in the proteins that make up life, and the type of amino acid, along with the order in which the amino acids are linked up, determine the shape and function of a protein. As you might expect, amino acids link up using dehydration reactions, just like monosaccharides do when they link up to form polysaccharides. As the amino acids link together, ejecting water, a bond called a peptide bond forms. Peptide bond, a bond that links amino acids together in a protein. Now, unfortunately, it is impossible to give you a structural formula for a protein. You see, the average amino acid has about 20 to 40 atoms, and even the simplest protein of life contains 124 amino acids linked together. Obviously, then, drawing the structural formula of a protein would be quite a job. The situation would be even worse for the average protein in the chemistry of life, which contains several thousand amino acids. Even though it's possible to give you a structural formula for a protein, it is possible to give you some idea of what one looks like. Figure 5.8 is a schematic representation of a protein called ribonuclease or ribonuclease. What does this schematic mean? Well, each three-letter abbreviation stands for an amino acid. The letters LYS, for example, stand for the amino acid called lysine, while the letters GLU stand for glutamic acid. This schematic then tells you the type of amino acids and the order in which they appear in the protein ribonuclease. In addition, the shape that the amino acids are lined up in is roughly equivalent to the shape of the ribonuclease. Of course, since the paper is only two-dimensional, it is impossible to draw the entire three-dimensional shape. But what is drawn here is a reasonable approximation. Remember back in module number 1 when we promised to tell you why we think that abiogenesis, the formation of life from non-living chemicals, is impossible? Well, proteins are a part of the reason. In order for abiogenesis to work, proteins would have to be able to form from random chemical reactions. Without this happening, life could never appear because proteins are such a fundamental component of the chemical reactions that make life possible. It is our contention that the formation of proteins from random chemical reactions is impossible. You see, when assembled as shown in figure 5.8, those 124 amino acids form a protein, a ribonuclease, that performs a vital task in the chemistry of life. If the amino acids were to link up in the wrong order, or if even one of the amino acids were the wrong type, then the resulting protein would be unable to perform ribonuclease's job. Thus, if the first amino acid were GLU instead of LYS, the protein would no longer be ribonuclease and would be unable to do the job that ribonuclease must do. Think for a moment about the probability of such a protein forming by chance. Is it possible for such a molecule to form by chance from a mixture of lots of amino acids? Let's make it easy on ourselves and assume that the only amino acids in the mixture are the 17 types needed to make this particular protein. In fact, there are about 20 amino acids that are a part of the chemistry of life. But adding more amino acid types would significantly reduce our chance of forming ribonuclease. So, in order to make the outcome more likely, we will restrict ourselves to using only the 17 different types of amino acids that make up this molecule. Making this assumption, we can say that the possibility of forming a protein that has LYS as its first amino acid is 1 in 17. Those aren't bad odds at all. However, the chance of forming a protein with LYS as its first amino acid and GLU as its second amino acid is 1 in 17 times 1 in 17, or 1 in 289. Suddenly, the odds are looking less and less favorable. The probability of forming a protein whose first 3 amino acids are LYS, GLU, and THR in that order are 1 in 17 times 1 in 17 times 1 in 17, or 1 in 4,913. If you were to complete this calculation, you would find that the odds for making this protein by chance from a mixture of the proper amino acids is approximately 1 in 10 to the 152nd power. A 1 followed by 152 zeros. In order to illustrate just how ridiculously low these odds are, the probability for forming ribonuclease by chance is roughly equivalent to the probability of a poker player drawing a royal flush, the most unlikely hand you can draw, 19 times in a row without ever exchanging cards. Remember, ribonuclease is a simple protein. There are proteins in our bodies that contain more than 10,000 amino acids. Clearly, the idea that these proteins could form by chance is absurd, which makes the whole idea of abiogenesis very hard for a scientist to believe. Enzymes are a special class of proteins that act as catalysts for many of the chemical reactions that support life. For example, remember from our discussion of carbohydrates that in order to digest disaccharides and polysaccharides, animals must break them down into monosaccharides using hydrolysis. Well, it turns out that hydrolysis reactions like these are incredibly slow, and animals would die long before they could digest polysaccharides if it weren't for enzymes. These proteins are catalysts for the hydrolysis reactions, making them fast enough to allow the proper digestion of food. It turns out that most enzymes do their job based on the shape that the enzyme molecule has. The shape of a hydrolysis enzyme, for example, complements the disaccharide or polysaccharide that is trying to break down. As a result, the enzyme can attach itself to the molecule it is breaking down and help force the water between the monosaccharides, speeding up the hydrolysis reaction. This process is illustrated in Figure 5.9 for the specific case of sucrase, the enzyme that catalyzes the hydrolysis reaction that breaks down table sugar, sucrose, into its monosaccharides, which are glucose and fructose. As we mentioned before, when you eat sucrose, your body must break it down into its two monosaccharides, glucose and fructose, before it can be used. Of course, the body uses a hydrolysis reaction to do this. To speed up the hydrolysis reaction, let's begin there again. To speed up the hydrolysis reaction, the body produces an enzyme called sucrase. This enzyme has an active site that complements the shape of the sucrose molecule. As a result, the sucrose molecule fits snugly into the active site of sucrase, and sucrase can then make it easier for the water molecule to react with the sucrose. Once that happens, the enzyme has not changed, but the sucrose has been broken down into glucose and fructose. The sucrase, since it has not been changed, is free to catalyze more hydrolysis reactions so that the more sucrose molecules can be broken down. Because the sucrose fits into the sucrase like a key fits into a lock, this view of how an enzyme works is often called the lock and key theory of enzyme action. Of course, if you think about it, a key can only fit one lock. Thus, sucrase is best used to catalyze the hydrolysis of sucrose, not some other disaccharide. After all, the shape of another disaccharide will be different and will not fit into the active site of sucrase as well as sucrose does. Thus, you often find that a given chemical reaction in the body is associated with a given enzyme. If a person does not have a particular enzyme, he or she might be able to digest most carbohydrates fine. But there will be one carbohydrate that he or she cannot digest well. This is the case with people who are lactose intolerant. These people cannot digest milk or milk-based products. As a result, they can get sick by eating or drinking dairy products. This happens because certain cells in their bodies are defective and cannot manufacture the enzyme necessary to catalyze the hydrolysis of lactose, the disaccharide in milk. Thus, They can digest many carbohydrates just fine, but they cannot digest lactose. As a result, they cannot drink milk or eat many dairy products. Now, it turns out that enzymes, because they're so complex and because their function is shape dependent, are quite fragile. Enzymes are easy to destroy, as can be illustrated in the following experiment. You should have seen that the Jell-O in the bowl with nothing added gelled as you would expect, as did the Jell-O in the bowl that had heated pineapple juice added to it. However, the Jell-O in the bowl that had room temperature pineapple juice in it should not have gelled. If it did, you did not use fresh pineapple. Why did this happen? Pineapple contains an enzyme that stops the reaction which causes Jell-O to gel. As a result, when pineapple juice is added to Jell-O, the Jell-O cannot gel. As is the case with most enzymes, however, this enzyme is very fragile. The heat that you added to the thick, pulpy pineapple mixture in the pot was enough to destroy the enzyme. And that's why the Jell-O in the bowl with the heated pineapple juice was still able to gel. This should give you an idea of how fragile enzymes are. In fact, enzymes are so fragile that most food processing destroys them. That's why the experiment called for a fresh pineapple. In processed pineapple juice, canned or frozen, the enzyme used in this experiment has been destroyed by the processing. Interestingly enough, pineapple also contains enzymes which can break down some of the tissue in your gums. That's why your gums may bleed when you eat a fresh pineapple. Because enzymes are so fragile, they break down soon after they are formed. Even in the fresh pineapple that you used in the experiment, much of the relevant enzyme had already broken down before you used it. However, even a tiny amount of enzyme is enough to see the effect that you saw in the experiment. So even though a lot of the enzyme in the pineapple was gone, enough was left to make the experiment work. Since enzymes break down soon after they are formed, your body must continually produce more and more enzymes just to replace the ones that are breaking down. That's why it's important to eat protein in your diet. The protein that you eat gets broken down into its constituent amino acids, and those amino acids are shipped to the cells in your body so that they can produce the enzymes that your body needs. DNA. Our discussion of the chemistry of life would not be complete, of course, without a brief description of DNA, the molecule which forms the basis of life. If you thought proteins were complex, you haven't seen anything until you have studied DNA. To begin your study of DNA, examine the schematic representations given below. Deoxyribonucleic acid, or DNA, is a double chain of chemical units known as nucleotides. These two chains twist around one another in the double helix that is so familiar to most people who have studied any amount of biology. The nucleotides that make up these two chains are composed of three basic constituents: deoxyribose, a simple sugar that contains 5 carbons, a phosphate group, an arrangement of phosphorus, hydrogen, and oxygen atoms, and a nucleotide base. A nucleotide's base can be one of 4 different types: adenine, thymine, guanine, or cytosine. In the lower part of the left side of the figure, you can see that the phosphate groups link to the deoxyribose units, which support the bases. The 2 nucleotide chains are held together because the bases link together in a process known as hydrogen bonding. Hydrogen bond: a strong attraction between hydrogen atoms and certain other atoms, usually oxygen or nitrogen, in specific molecules. Hydrogen bonding is actually a very complex process that you'll learn much more about when you study chemistry. For right now, you just need to realize that the attraction between the atoms in hydrogen bonding is about 15% as strong as the attraction between 2 atoms that have a true chemical bond linking them. Thus, the hydrogen bonds in DNA are strong enough to keep the 2 chains together in a double helix, but they are significantly weaker than a true chemical bond. Since they are weaker than a true chemical bond, it is rather easy for the 2 helixes in DNA to unravel. That, as we will learn in the next chapter, is very fortunate. Hydrogen bonding is highly dependent on the molecules involved, and as a result, only certain nucleotide bases can link together using hydrogen bonds. The nucleotide base adenine can only the nucleotide base adenine can only hydrogen bond to thymine. It cannot hydrogen bond to cytosine or guanine. In the same way, thymine can only hydrogen bond to adenine. Likewise, cytosine can only hydrogen bond to guanine and guanine to cytosine. As a result, DNA is often pictured in a very simple way, as shown below. In this figure, T represents thymine, A adenine, G guanine, and C cytosine. The ribbons in the figure represent the backbone of the DNA. Notice that T's are only linked to A's and C's are only linked to G's and vice versa. This represents the fact that only adenines and thymines can link together, and that only cytosines and guanines can link together. Now you can finally learn how DNA stores all of the marvelous information that it uses as the instructions for making life. Just as the entire English language can be reduced to sequences of dots and dashes in Morse code, all of the information necessary for life can be reduced to sequences of nucleotide bases in a DNA molecule. Cells, as we will learn in the next module, have chemical machinery that decodes the sequences of nucleotide bases into instructions for what structures need to be built and where to build them. Isn't that marvelous? Of course, that's not the end of the story. One of the most interesting questions you can ask about DNA is how much information can be stored there. After all, modern technology has gotten pretty good at storing information. We can now make handheld computers that hold more information than a whole shelf full of books. How does that compare to the amount of information that DNA can hold? It doesn't even come close. Calculations indicate that if we were to write out all of the information that can be stored in a sample of DNA the size of a pinhead, we would end up with a pile of books that is 500 times higher than the distance from the Earth to the moon. This should tell you something about the incredible power and wisdom of the one who designed DNA. I'll see you next time, friends, for Module 6. Bye-bye for now.